Well, good morning, everyone. My name is Li Ping. I'm a postdoc working in Vijay's group and also Todd Martinez's group here at Stanford in the chemistry department. I just want to say that it's really exciting to see all of these folks from all over the world to come here to learn about molecular dynamics and analysis of molecular dynamics simulations. And now I'm going to talk about classical molecular dynamics simulation, kind of an overview and introduction, just to see where kind of like MD simulation takes its place in the overall universe of research. Here's kind of the overall structure of the talk. I will introduce molecular dynamics first, followed by some of the methods, including force fields and integrators and so on, and then some of the interesting applications that will be available to you after you learn how to effectively run and analyze molecular dynamics simulations. Molecular dynamics kind of fits into this discipline of physical chemistry, although it's also found in condensed matter physics and material science. And the overall idea is that chemical processes are governed by physical equations. And so physics helps us to understand these chemical processes. They range from chemical synthesis, which allows us to make lots of things, including synthetic fabrics that go into dresses, as well as bulletproof vests. You can also carry out catalysis, which means accelerating chemical reactions by many orders of magnitude, such as creating fuels, like in a catalytic cracking, or reducing pollution. That's the catalytic converter there. Another big area of physical chemistry is studying molecular interactions. So the interactions of electric potentials with molecules gives you electrochemistry, and interactions of light with molecules gives you photochemistry. For example, if you don't want to look like DJ Pauly D from Jersey Shore here, you put on sunscreen, right? And sunscreen kind of absorbs UV radiation. So that's kind of a photochemical thing. And then when molecules interact with other molecules, those are called non-covalent interactions, which are really important in protein folding or drug binding and conformational change and things like that. So not only is molecular dynamics kind of a subset of physical chemistry or condensed matter physics, it's also theoretical. And there's a really important interplay that goes on between theoretical and theoretical and experimental chemistry. And this is kind of like the typical interaction between a theorist and the experimentalist. And I think this picture is kind of funny because it's the theorist that's wearing the glasses here. The experimentalist is not wearing eye protection. But a collaboration might look like this. The experimentalist finds some interesting observations, and their experimental instrumentation or their measurement leaves them kind of in need of a more atomic resolution explanation. And that's what the simulations are for. The simulations give us insight into atomic scale processes happening at essentially as fine of a resolution as you want, because it's happening in your computer. So that gives us kind of like some insight into what's going on. The experimentalist can verify the approximations that the theorist has made, and then the theorist can also even predict new things using their simulations for the experimentalist to test. So that's kind of how this experimental theoretical interaction works. And a lot of the more difficult problems in chemistry and physics requires both experiment and theory in order to solve. So this is kind of like a picture of photosynthesis. These light harvesting proteins capture light. This exciton will now travel into the photosystem 2, where it separates into an electron and a hole. And then protons get reduced kind of to the equivalent of hydrogen on one side, and then water gets split to make oxygen on the other side. In order to understand this process, a lot of experiment and simulation were both involved, including resolving, say, the structure of the oxygen evolving center. This is an X-ray crystallography study, but actually a lot of theoretical methods go into interpreting these X-ray measurements. So what are the physical theories that go into molecular dynamics simulation? It's really kind of a combination of quantum mechanics and statistical mechanics. Quantum mechanics tells us how the atoms interact because the electrons in a molecule are completely described by solving Schrödinger's equation. The interesting thing about Schrödinger's equation is that you can't solve it analytically for any system containing more than one electron. So in most situations, approximations are used. And statistical mechanics comes into play because what we're interested in studying often occupies a vast population of states, and we're trying to get kind of some information out of this very wide ensemble of states. So this partition function provides the probabilities of all the different states that your system can be in, and this high dimensional integral, which is also often not, you can't evaluate it analytically. You evaluate it by sampling, which comes from computer simulations. So I'm going to very briefly talk about Schrödinger's equation and see how it actually ties into molecular dynamics. You might find it interesting as well. So all of the ground state quantum chemistry, which tells you where the electrons are in a molecule, comes from solving this time-independent Schrödinger's equation here, where H is the Hamiltonian operator, psi is the wave function containing all the electron coordinates, and E is like this energy eigenvalue. So what this is telling us is that H, this operator, so an operator takes a function and gives you another function back out. This Hamiltonian operator, when applied to the wave function, gives you the wave function back again, but multiplied by a number, and that's the energy. And the various components of this Hamiltonian operator represent interactions or energy terms, representing, say, the kinetic energy of the electrons, the interaction of the nuclei with each other, the nuclei with the electrons, and the electrons with each other. So by solving this equation, this gives us the electronic wave function, which tells us where the electrons are, and then because the electronic energy is kind of this function of where the nuclei are, that gives you a potential energy for the atoms to move. So in doing all of this, we kind of invoke the Born-Oppenheimer approximation and separate the timescales of nuclear and electronic motion, so what you get is that the potential energy of the system is a function of the atomic positions, because you assume that the electrons are always kind of in their electronic ground state, and that provides this potential surface for the atoms to move on. So because Schrödinger's equation is nearly impossible to solve for any but the most trivial systems, we use approximate methods to get our approximate solutions. So the thing that makes this problem difficult is the electron repulsion, which makes it so that we can't separate our differential equation in the usual way that we solve systems of differential equations. So the first approach is that use approximate wave function forms, and that's called quantum chemistry. So this is one such approximate wave function called a Slater determinant, and if you try to, and this contains a bunch of adjustable constants in it, so it's kind of like this ansatz, or an educated guess for the functional form of our wave function. By using this approximate wave function form, we get an approximate solution to the Schrödinger equation. That's quantum chemistry. Another approach is instead of the wave function, you use the electron density as the main variable. This is an important discovery in the 20th century that says the electron density really contains all of the same information as the electron wave function, which is actually a very important result. So density functional theory just says the electron density contains all the information, and theoretical approaches that use the electron density have also been very successful in the past several decades. And the third approach, which is kind of what we're going to be focusing on in this workshop, is that you approximate the energy using empirical functions. So here you see that there's really kind of a close connection between these quantum chemistry methods and the molecular mechanics force fields that you'll kind of be familiar with. Because what the force fields do is that they do not treat they don't treat the electron degrees of freedom explicitly, right? You're never solving for where the electrons are, but it kind of gives you the sense that the potential energy that there is this potential energy function that depends on the atomic positions. So if you were solving for the electron degrees of freedom, the wave function would give you what the potential energy is, and if you are using molecular mechanics, then this empirical function called a force field tells you what the energy is. And this is kind of an overview of these computational simulations of molecules. And you can see that there's really a very wide range of these that span a wide range of calculation costs and amount of physical detail, where if you want more physical detail, of course you need a more expensive calculation. So the numerical solution of Schrödinger's equation, also kind of known as full configuration interaction, is way up in the upper right, and it's unfeasible for anything but the smallest systems. But it gives you the most physical detail, right? In order to study chemical reactions, we use quantum chemistry like Hartree-Fokker density functional theory. In order to study larger systems for longer timescales, which is kind of the main interest of this workshop, hundreds of thousands of atoms, maybe for microseconds to millisecond time scales. We use molecular mechanics. And you can also do these coarse grain simulations where individual particles represent more than one atom. And kind of this area is what OpenMM is specialized at, one of the software packages that are developed in the lab and that you're probably going to learn about these 2 days. So here's kind of some of the things that molecular dynamics can tell us. If you run a simulation with a force field, you can actually look at the time evolution of an entire protein. So if you start the protein off in an unfolded state, you see certain structure appears early while other structure appears later. So on the right side, I'm going to be showing you a very different simulation where a lot of molecules are just smashed together and react with each other. And on the right side, I'm going to be using kind of chemistry, a level of theory, where the simulation is going to be much smaller and much shorter than the one on the left, but you're going to see very different types of transformations. So on the right here, you can see that as the molecules smash together, they're all different colors because I'm labeling the molecules by the different species that they're forming. And as you can see, there's a very wide range of applicability of these types of simulations. Furthermore, analysis is really important in these types of simulations because, as you can see, there's a whole lot of information that's being generated. And how do we turn information into knowledge? And that's kind of where analysis methods like Markov state models come in, where each individual state represents like kind of a coarse grain conformational state of a protein, and it can make transitions between the states in this kind of network. So with that introduction, I will start to talk about the methods in molecular dynamics simulation, and it really starts with Newton's second law. So maybe to clear up a little bit of confusion here, no matter whether you use the electrons, no matter whether you use the quantum chemistry or density functional theory or a force field to get your potential energy, in this workshop we are mainly going to be talking about using Newton's second law to move the atoms. Okay, so the force on the atoms is given by the negative gradient of the potential energy. For us, this is going to come from a force field, but it can also come from an electron wave function. And knowing the force allows us to accelerate the atoms in the direction of the force. Of course, this is an approximate. Of course, this is also an approximation because atoms themselves are quantum mechanical, right? But we're going to move them according to the laws of classical mechanics. The key approximations that we're going to be making here, that there's no electronic transitions, of course. It's an approximate potential energy surface. We can never really get an exact potential energy surface. And we're using classical mechanics for the atoms. Okay, so now to talk about more details of the methods. So force fields are a topic that are very near and dear to my heart. They are potential energy functions that are really comprised of these functional forms that you can just write down on a sheet of paper, and empirical parameters that tell you how strong the interactions are. So these are what the bonded interactions look like. You could have something just as simple as a harmonic vibration between pairs of atoms, or an angle vibration between 3 atoms, or a periodic kind of torsion interaction between 4 atoms. Those are the bonded interactions. And between pairs of atoms that are not bonded to each other, you have non-bonded pairwise interactions where there might be an electrostatic attraction or repulsion at long range, and also this short-range repulsion that keeps the atoms from going on top of each other. Now, because a force field contains these parameters that say how strong the interactions are, you are free to create as unrealistic a simulation as you want, right? Just by picking parameters that are not accurate. So in order to have a simulation that really corresponds with reality, force field parameterization is a major problem. So force fields are typically parameterized to compensate for their simplified description of reality. In the bottom, I've shown you a few pictures of models for water, so force fields for water, and they're represented using 3 particles representing the atoms, or 4 particles if you want like a more detailed description of the electric charges. You can even go to higher sites, but the main idea is that the physics is incomplete. Okay, so first of all, these water molecules cannot dissociate. They're usually rigid, so they can't even vibrate. They can just kind of like rotate and move around. And furthermore, they follow the laws of classical mechanics. So, but you can recover a lot of kind of the correspondence with reality through parameterization, because the force field parameters are kind of responsible for effectively capturing the interactions that is not explicitly captured by the approximate details in your force field. So in many cases, the force fields actually exceed the accuracy of the electronic structure methods just because they include some amount of experimental data in their parameterization. It might even allow them to have more predictive power in certain areas. So, an interesting question might be, how detailed does the function need to be in order to describe reality? Because in reality, we know that a water molecule might be kind of a complicated object. It's got nuclei and it's got an electron distribution. This is really a wave function with electron correlation and stuff. And in order to represent it in a force field, you can do certain things like put a point charge on each atom. That's what the amber fixed charge force field does for all of its proteins, and this is how also the TIP3P water model works. But you can also go more complicated than that and put higher order multipoles on each atom, and even include electronic polarizability. So as you can see, the thing on the right is now a more accurate description of the thing on the left. But a more accurate description also comes with more complicated equations. So your point charges look like this, whereas your multipoles kind of look like this. And when you have more complicated equations, that means not only is it going to be harder to code up on the computer, but it's also going to take a lot longer for the computer to evaluate. So, so an important question in all of our research is to choose the model with the amount of detail that's appropriate for our application. And now I'm going to be talking a little bit specifically about the balance between solute and solvent force fields. So, so one must be careful to avoid force field bias in the simulation results. If your solute interactions are too strong, then it's going to unfold your protein, whereas if your protein interactions are too strong, it's always going to fold. So this force field bias is kind of a source of error in our simulations, right? That's an important thing to keep in mind. And the final note that I want to say about force fields, I think, is to show the many levels of detail of the models that are available to you. So in many cases, you can actually use different levels of detail for your solvent and your solute. And because the most common solvent is water, there's many different ways you can model water. And that includes these implicit solvent models where there's no solvent particles, like Cosmo or generalized Born, coarse-grained particles where each molecule is represented by a single particle and not three. Maybe the most familiar explicit solvent all-atom models like TIP3P, and then going up to the higher order multipoles. So as you go to the right, you get more physical detail, but also a higher computational cost. Okay, so now I will talk about the ways you integrate the equations of motion and some other methods in molecular dynamics. You need to, so now that you have a force field which gives you the potential energies and the forces which give you the acceleration, you need ways to integrate Newton's equations. And the solution of Newton's equations in kind of these complicated potentials is not an analytical solution. We need to do it numerically, so that's why we have molecular dynamics integrators. And the simplest thing we can think of is probably to just change the positions based on what velocities we currently have, and then change the velocities based on the accelerations that we currently have. And that's called the Euler method. But the Euler method is actually not a very good integrator at all. It's unstable. So there are other more sophisticated methods available for integrating the equations of motion, such as the velocity relay method, which is very commonly used. And I think OpenMM uses either this or a leapfrog method, which is very close to velocity relay. And one important thing to realize about these integrators is that they are often is that the time step in the integrator is often what limits the length of our simulations, because the processes that we want to study might occur on microsecond or millisecond or even longer time scales. But the time step of these integrators is extremely short, because the fastest degree of freedom in these simulations might be the vibration of a chemical bond, which is like 10 femtoseconds. So typically, molecular dynamics integrators have a 2 femtosecond time step, meaning if you want to simulate a microsecond process, you need almost 1 billion time steps, which gives rise to a pretty high computational cost. That's how you want codes that are fast. And another interesting thing to note about molecular dynamics simulations is that they are chaotic. By chaotic, that doesn't mean completely random, but it means that if your initial conditions are just a little bit different, then your final result might be very different. It's kind of like dropping a tennis ball on kind of like cobblestones. You know, you have no idea where it's going to bounce, right? As a kind of a little aside here, people have integrated Newton's equations for planets, not molecules. And what they did here is is using the gravitational potential. They integrated Newton's equations to predict where the planets are going to end up in a few billion years. And they predicted that if they change their initial conditions by just a few centimeters, okay, then they'll end up with Mars crashing into Earth in like a billion years or something. So that was published in Nature, and I thought that was kind of interesting. Okay, so now let's talk about some more topics that are specific to molecular dynamic simulation. Something that's really important that we also need to keep in mind is the boundary conditions. So typically, the system that we want to simulate is in some environment. You might have an ion surrounded by a solvent, or you might have a membrane protein in a lipid bilayer. But because of our limited computational resources, we can only study systems of finite size. And furthermore, if you simply like drew a wall around your system, the edge effects will be significant enough that you want a way to simulate a system of finite size without edge effects that are too significant. So the typical way to do this is to invoke periodic boundary conditions, and that means that when your simulation is, say your simulation is represented by one of these boxes, the equations that are actually being integrated kind of contain these periodic replicas of the boxes. So the atoms kind of on the edge of the periodic box are actually interacting with the periodic images of the atoms on the other side of the box. And there are ways to treat long-range interactions in periodic boundary conditions. That's what the particle mesh Ewald method is for. And some important considerations is that you don't want your molecule to interact too closely with an image of itself. You might kind of like get spurious results and stuff. So periodic boundary conditions are another important consideration. Now let's talk just very briefly about statistical mechanical ensembles. Kind of this little table that I'm showing you here, the idea that I want to convey is that every like kind of property on the left is kind of conjugated with a property on the right. So when you run a simulation, you can choose to conserve the particle number, the volume, and the energy. That would be kind of like the most basic or NVE type of simulation. Or you can choose to not conserve the energy, but kind of have an average simulation temperature instead. That means the algorithm is going to put some energy into the system or take some energy out. Or you can do an NPT simulation where your system will kind of fluctuate around an equilibrium size depending on the external pressure that you're applying. Or you can use a chemical potential where particles are actually kind of going in and coming out of your system. And so these are just kind of the statistical mechanical ensembles that are available. And I've kind of colored them by how difficult they are. So NVT simulations are actually kind of like the easiest thing. So constant pressure and energy conserving simulations are a little bit more challenging. And then simulations with kind of like particles that go in and out are the hardest. Okay, so let's talk about how we control these statistical mechanical ensembles. So first of all, conceptually, energy conservation is really the easiest. You just need to integrate your equations of motion as accurately as possible. The reason why I said it was challenging is that in practice you need to be very careful in order to do it right. But say if you want to run a constant temperature simulation, that means there's going to be an algorithm that will make sure that the velocities of your particles, which correspond to the instantaneous temperature, are kind of fluctuating around an equilibrium value that you set at the start of the simulation. So this is the kind of relationship between the kinetic energy of your particles and the temperature, and a very simple algorithm to make sure that your particles are kind of like at the correct average temperature is the Berenson thermostat, which just scales your velocities down if they are moving too fast and scales them up if they're moving too slow. However, this doesn't produce the correct canonical ensemble. So the statistical mechanical ensemble that corresponds to a particular temperature, this produces a distribution that is far too narrow. So the Berenson thermostat is actually not a very good way to go. And a better thing might be the Anderson thermostat, which periodically randomizes the velocities of your particles. So the Anderson thermostat will give you the correct canonical ensemble, but because it's randomizing the velocity of your particles, if you're trying to calculate any kinetic properties, then you have to be careful because the thermostat is going to affect your kinetic properties. Now let's talk about some other ensembles that are not strictly molecular dynamics, but kind of closely related to it. For example, Langevin dynamics is it's a lot like Newton's equations of motion, except you have a friction force which slows your particles down, and kind of a random force that perturbs their velocities randomly. And Langevin dynamics is kind of meant to represent, say a particle being buffeted on all sides by smaller particles, but it's also another way to carry out temperature control in a molecular dynamics simulation. So in fact, a number of you might even be using a Langevin integrator when you're running a molecular dynamics simulation, just because it's a pretty good way to provide temperature control. You can also do a Monte Carlo simulation where there really is no concept of time at all. In that case, you just have a series of, say, pre-programmed or random moves, which you either choose to accept or reject using this metropolis criterion. And that is a way to also produce a canonical ensemble without actually integrating any equations of motion. So using Monte Carlo sampling, you don't need any forces, you don't need an integrator, but you, of course, don't get any kinetic properties either. So now that I've told you all about like the methods that exist in molecular dynamics, what are the challenges that are still there? This is kind of a listing of the challenges that are still there. I stole this image from VJ. It's just kind of a picture of the kind of the time scales that are accessible to us and the time scales that we really want to do. And there's still like orders of magnitude that often separate us from the easy simulations we can run and the processes that we actually want to study. So sampling is a big problem, right? You need to efficiently sample the ensemble and have code that is fast enough to simulate a long time. So that's also kind of an algorithms and computer science thing because you need to write your code quickly. You could sample for a really long time and get answers that don't make sense if you have a force field that's not accurate. So accurate force fields are an important problem. And then if you generate, say, a terabyte of data, it's sitting on your hard disk somewhere. But that doesn't mean you automatically generate the science, right? You need to analyze the data. And that's also something that we're going to be learning about in this workshop. So with that, I'm going to go into some applications of molecular dynamics. So let's talk a little bit about, say, applications. So I'm going to show you some quick examples of things that have kind of been going on in research here. So one of the things that the Pandey Lab has been doing for a long time is studying the protein folding problem. So that includes predicting the structure of a folded protein, and also the pathways by which the protein goes from an unfolded structure to a folded structure. So structure prediction is like, can we accurately predict the structures from kind of starting from an unfolded structure. In many of these cases, the structures are already known at the start of the study, but if you run the simulation and it kind of goes to something that overlaps very well with the experimental structure, then you're happy. This kind of a D. Shaw like science paper that I think a lot of people paid attention to. But also these folding studies kind of help us understand proteins that don't have any structure at all, like amyloid beta, which is really important for Alzheimer's disease. And in addition to structure prediction, we might look at protein folding mechanisms. So is there a single preferred pathway, or are there many pathways? So if you have an extremely powerful supercomputer that costs $1 billion, it's probably not $1 billion, but you can run a very long simulation trajectory and see what that trajectory does, because that's the data that is available to you. And you'll find that the folding follows a certain pathway. But if your computational resource is distributed, which is kind of what happens at Stanford, we have the Folding at Home project has donors that kind of donate their computer time, and as a result, you get many very short simulations, right? Then the approach there is to use an analysis method like MSM Builder to take your many short simulations and coalesce them into a single model. And that gives you a picture like this, where you have these Markov states and they make transitions, And as you can see, you get different pictures out of these two methods. You either get a single trajectory that shows you that there is a pathway that the protein folds this way, or you get this listing or this map of states and transition rates between them that tell you that there's really lots of different ways that the protein can go from unfolded to folded. Now, for proteins that are too big to fold, to fold in the computer, you can still study these proteins that already have crystal structures and study how they function. And that's kind of what conformational change is. Say we might like to understand signal transduction from the outside of the cell to the inside. And that is a project that happened in the Pane group also, studying the activation mechanism of G-protein coupled receptors. So this is kind of like a picture of a G-protein coupled receptor in the blue, embedded in a lipid bilayer. And what happens is that first this signaling molecule binds to the outside. Okay, the signaling molecule doesn't go to the inside. The membrane will keep it from going inside, but it will kind of bind in here. That will cause a change in this receptor geometry. So the change in the geometry is big enough that it actually propagates to the inside of the cell. So I like to think of the GPCR kind of like a doorbell. You know, signaling molecule binds to the outside and the signal goes to the inside. It causes the G protein to bind on the inside, and then this nucleotide is, which is kind of represented in USA colors. The nucleotide is released and it initiates a signaling cascade. So how this mechanism, how this activation mechanism works, could be a very interesting problem because first, both the active and the inactive structures have been crystallized. That's what Brian Kobilka won the Nobel Prize for. And as you can see, the active and inactive states really have a very subtle geometric difference, at least from a non-specialist like myself. And so we might like to know whether there exists any intermediate paths or just the kind of the finer details of this activation pathway. That's kind of what happened in the Pane group. So by running a lot of molecular dynamics simulations and using Markov state models for the analysis, there were intermediates on the G protein coupled receptor activation pathway that were identified. So that was kind of an interesting project. Another thing you might want to use molecular dynamics for in a biochemical setting is to calculate these protein ligand binding free energies. So how strongly does a molecule stick to a protein? This could be like a drug molecule or something. You might want to understand why a particular small molecule drug works so well, or you might even be working at a drug company and you have this molecule you really like that you want to see how well it works. So these simulations involve the ligand molecule repeatedly visiting the binding pocket and leaving again. So it's a pretty expensive simulation, but it could give you more accurate results. And then as kind of, I think, one last application, I want to talk about one of my research projects that I worked on here. Because I work a lot on force field development, one of my projects was to try to make a water model. Okay, and the water model that I focused on was derived from this amoeba polarizable model that came out 10 years ago. But we're going to kind of neglect one of the key interactions in the model, which will make the calculation a lot faster. But because it neglects a piece of the physics, we need to reparameterize it. Okay, so we first take out a big piece of the amoeba model, and then we want to reparameterize the model. And the method that I developed for parameterizing force fields is called force balance. This is kind of a schematic of how the optimization cycle works. And basically, the program will allow you to put in experimental data and ab initio calculations at the same time. So the model that comes out at the end is hopefully gives you an accurate fit to both the experimental data and the ab initio calculations, so it's more predictive. And this is kind of the result of the fit. So the blue curve here is the more expensive amoeba model from 10 years ago. If you omit the important interaction from the amoeba model that makes it cheaper, you get this dark green curve. So these are densities of water with respect to temperature. So this is really an important interaction in the amoeba model that you're taking away, because once you take it away, the water model behavior kind of becomes unphysical and you get this dark green curve here. But once we refit the parameters with force balance using this density profile as fitting data, you pretty much reproduce the experimental result exactly. So the model that you get at the end is not only cheaper than the original amoeba model, it's also more accurate. This is kind of a representation of what the quantum mechanical forces and the force field forces look like. Say one set of vectors represents like some high-level ab initio calculation, and the other set of vectors represent the force field. It gives you an idea for how these force vectors agree. Well, to really test whether a force field is good, you need to try to simulate properties that were not part of your training set. So one of the things we did is that we calculated the phase diagram of water and the high pressure ices, and that's carried out using simulations like this. So this is a two-phase simulation where you have ice on the left and liquid on the right. And this particular movie is not ordinary ice. It's actually one of the forms of high pressure ice. And as the simulation freezes, you see there is a trend in your order parameters. So what you want to see is kind of a trend in this graph and the histograms, and kind of these molecules will tend towards freezing. And after running many of these simulations, it allows us to determine the melting points of the different phases of ice and allow us to construct a phase diagram. And because the phase diagram was not a part of our fitting data, it was a pretty good result for us to find that the phase diagram predicted by our model was in agreement with experiment. Okay, so with that, to actually run these simulations, there are several software packages that are available. The one that you guys are going to be learning about mostly in this workshop is called OpenMM. This is kind of a summary of several software packages, not all of them. A lot of you might have used one or more of these packages, so have I. And I would say that one of the ways that OpenMM might be different from the other pieces of software is that first, well, it can run on GPUs and on a very kind of wide array of platforms, and it's also very flexible. It gives you a very flexible programming interface in order for you to do kind of a specialized research problem. And another good thing is that it is a free and open source software. So with that, I'd like to conclude my talk. If you'd like to read more about molecular dynamics simulation, I would probably recommend these two texts. These are probably the most widely read textbooks for molecular dynamics simulation, I guess. And yeah, here's some papers that I looked up to, for some of those applications. Yeah, that's it. Thanks. I'd like to take your questions now.